Muy buenas tardes a todas y todos. En nombre del Instituto de Investigaciones en Matemáticas Aplicadas y en Sistemas, les doy la más cordial bienvenida a esta conferencia que forma parte de la visita de una de las científicas más influyentes de nuestro tiempo, la doctora Ingrid Daubechies, quien hoy nos honra con su presencia. Agradecemos la asistencia de las autoridades que nos acompañan, de todos los presentes, así como la de todos aquellos que nos siguen de manera remota a través de la plataforma Zoom. Antes de comenzar, quiero destacar la importancia de este momento. La doctora Daubechies ha revolucionado el campo de las matemáticas aplicadas con su trabajo en compresión de imágenes mediante ondoletas u ondículas, una contribución que ha transformado múltiples áreas del conocimiento y de la tecnología. Por cortesía con la doctora Daubechies, continuaré la presentación en inglés. It is a great honor to introduce Professor Ingrid Daubechies, the James B. Duke Distinguished Professor at Duke University. Professor Daubechies is a globally renowned leader in the mathematical theory of wavelets, and her pioneering contributions have profoundly impacted various fields of science and technology. Among her many groundbreaking achievements, she is best known for discovering compactly supported continuous wavelets. This contribution has been pivotal in detecting gravitational waves, revolutionizing signal and image processing, thereby laying the foundation for digital cinema and the JPEG format, providing valuable tools for art historians and conservators, aiding in establishing the authenticity and age of masterpieces by artists such as Vincent van Gogh and Rembrandt. Her research spans a remarkable array of fields, including orthogonal transformations, harmonic analysis, and multi-resolution. Her work has had a profound influence on machine learning, bioinformatics, artificial intelligence, data science, and medical research, among many other disciplines. Professor Daubechies has also achieved many remarkable milestones. She was the first female full professor of mathematics at Princeton University. She remains the only woman to have served as president of the International Mathematical Union. Her many accolades include the National Academy of Sciences Award in Mathematics, the Nemers Prize in Mathematics, the Princess of Asturias Award, the Wolf Prize. She has received honorary doctorates from 8 universities, including our Universidad Nacional Autónoma de México in 2022. Today, we are truly honored to welcome Professor Daubechies, a scientist of extraordinary caliber whose work continues to shape our understanding of mathematics and science. On behalf of IIMAS and everyone present, I would like to express our deepest gratitude to Dr. Daubechies for sharing with us her invaluable knowledge through her talk entitled Discovering Low-Dimensional Manifolds in High-Dimensional Datasets. Demos la bienvenida a la doctora Daubechies.
Yes. Okay. Great.
Behind you. Oh, okay. It's better for the photos.
Okay. Okay. So thank you very much for this beautiful introduction, and to all of you for coming. I'm so glad to be here. When I came in 22, there was no time for me to stay longer and to give a presentation. So I'm glad to be back. And I apologize for not being able to speak in Spanish. I can just say hello and order a beer, and that's it. Okay, so a manifold or a variety may, and in many cases have the things, data sets of interest to us have manifolds of smaller dimension than the dimension in which the data are presented at first. And this is just a caricature of that. So it's even though points on this little contraption would be given by 3 coordinates, it's clearly only a 2-dimensional structure. So the idea is what we're going to do is try to recognize from the data the dimension it has and analyze things on those data in the dimension in which they're working. So if you look at these, then the forget what they're called, these little things that are sewn to the surface are all flat. And together, their union shows that, in fact, we're following a curved surface. So we're doing something similar, but computationally. So if we take this two-dimensional surface, then if we take only local points, they will lie approximately in a plane, in the tangent plane. We'll be very close to a tangent plane. So we look at these little paillettes on it, and if we have many of them, then they will follow that surface. And in our data set, if points are close together, then the distance that we measure between them in the ambient high-dimensional space is fairly close to the distance on that manifold, because we're so close to the tangent plane. But large distances cannot be trusted. Nevertheless, you can get to large distances on the data manifold by putting many of these little paillettes together, following every time the surface. And so small distances are more reliable. And this you can exploit computationally by saying that, well, the chord length is very close to the distance on the manifold, and that's very close to, if you pulled back everything to the tangent plane, to the distance you would find on the tangent plane between the pullback point and the touching point, the tangent point. And we're going to exploit that to say that we can try to diffuse over small distances on the manifold by using the same expression for the heat diffusion kernel that we would use in the high-dimensional space. That's a very remarkable thing about the diffusion operator, that its kernel looks the same in all spaces. I mean, the normalization is different, but all Euclidean spaces have the same expression for the kernel of diffusion. So we want to diffuse on the manifold of the data, but for small distances, we can use the diffusion expression on the ambient set, but we'll only diffuse from data point to data point. So if we have our data point, and we have a way of computing a dissimilarity distance, some distance, and we're going to wave our hands and say, okay, we'll treat it as Euclidean because, well, we don't know better. So then we can use that to build the diffusion kernel for the kernel for diffusion in Euclidean space. And the T here, oops, I think I have, yes, the T here is the time of diffusion. It measures how far we diffuse. So we will choose this T fairly small because that means that we look very locally. But we, of course, we have diffusion here and there and there and there and there. So what that means is that if we look at the whole matrix and we apply it several times, hopping small distances each time, it's like hopping over the whole manifold, going longer distances. So the fifth power of this matrix would give us something that manages to go from there to there. So I said T needs to be sufficiently small, and we'll have to come back to that. We also have to worry about normalizing. Well, we are going to use, we have to worry about the fact that sample points are not equally distributed, uniformly distributed on our surface necessarily, so we may have to adjust for that. But one adjustment is already done by realizing that what we build is a coarse approximation to a diffusion kernel, to a random walk. And so it should be, have the properties of a random walk, of a probability. So the sum of the entries with our unknown normalization should add up to 1, and that of course defines this NT. And you see it takes in consideration many things. It already accounts for the fact that some points may have more neighbors than others. I mean, it corrects for that. It also corrects for the dimensionality. Normally, the constant in front would express the dimensionality of the data set, of the space. And we work in a high dimensional space, so we would use that dimension. But we're going to normalize, taking into account that we use the points on the data set only, and that automatically will give us a normalization adapted to the dimension of the data set. So, in practice, what that means is that you build your matrix, and then you build a diagonal matrix having these sums, and you take the inverse of that in front. And so now you have a random walk defined on the graph, which you hope is a candidate for a discrete approximation of diffusion on the manifold of the data, of which the data points are sampled. And that also, turns out, gives us a good idea for choosing T. This method is not a method that I proposed. It was proposed by Belkin and Yogi. It was exploited a lot by Stéphane Lafont, Ronald Koifman, Mauro Maggioni, and many others. And most people who apply these diffusion methods to explore data sets say that choosing the time, diffusion time, is more of an art than a science. Because if you choose T too small, then, well, nothing has time to diffuse. You just get the identity matrix. And not very interesting, and will not tell you much about your data. If you choose T too large, then in the exponent here, if T is too large, then distances that are fairly large will give an exponent that is small, and with this minus sign means that this expression becomes sizable even for large distances. And you don't want to trust large distances. You only want to diffuse a small distance, so you need to T sufficiently small. So you need to take T not too small, but not too large. And one way of deciding whether you're doing that is, again, use this physical intuition of diffusion. I mean, if you truly had a diffusion operator, then that diffusion operator would obey the semigroup law. So one thing that you can do, and we have found that this works very well, is to take for different T, compare the square of the random walk, of the proposed random walk you define, with the random walk you get for replacing the T by 2T. And those two should be close to each other for it to be something that you want to interpret as a diffusion. And so in her PhD thesis, Shan, my former student Shan, who's now in Denmark, explored this. And for instance, for some standard data sets, so for instance, for this data set, which depending on what T you choose and the embedding that from the diffusion matrix and spectral analysis, which we'll come to later, from those embeddings, you can get this picture or this picture. And clearly, what you'd like to know is that it's really that picture. It's a one-dimensional data set that has been scrolled up in 3D. And one way of seeing that is to look at the semigroup error. So this error between WT squared and W2T that I was showing you, and plotting it as a function of T. And the one that is the best actually comes out as indeed the best. For smaller T, you're not learning enough of the manifold and your error is big. For larger T, you go, well, of course, if you go for T very large, then you're just computing all the same matrices and it satisfies, again, the semigroup law, but it's, again, without any content. For this Swiss roll data set, another standard, these are different embeddings you might obtain from this diffusion method, depending on the choice of T. But again, if you look at what the different choices correspond to in the semigroup error, the one that you really want, the one that's very close to a square, is the one that corresponds to the lowest. So we systematically use this, and it works very well for us. So that's already a first method. If you're interested in diffusion methods, even if you do completely different things from what you do, I would recommend that you use this rule of thumb to choose your parameter. Okay, so we have found this approach to have a candidate for discrete approximation. The idea then is that we are going to use this to reparameterize our data set. So this matrix, which you build as a symmetric matrix, is you can make a spectral decomposition, and of course the whole thing is noisy. So what that means is that you're going to trust the dominant eigenvectors, the eigenvectors which have eigenvalues closest to 1, and as the eigenvalues go down, you can trust the eigenvectors less and less. They are completely dominated by noise at some point. So typically, if you, the spectral decomposition, because it's a spectral decomposition of a diffusion matrix, powers of the eigenvalues give you a diffusion over another time. And so you can immediately build, you don't have to, in the spectral decomposition, compute powers of the matrix. You can just compute powers of the eigenvalues. And that then, what we're going to do is we're going to say, let's take only the first capital L dominant eigenvectors and use those to reparameterize the points. So every point I is going to be parametrized by taking the value in that point of the different eigenvectors up to capital L and weighing them with the eigenvalue up to a certain power. And then we can define a distance, a diffusion distance, by taking on this parametrization in this capital L dimensional space the corresponding Euclidean distance. And that's what's done when people do diffusion distance. So that's one thing that I will be applying. And as I said, this is a method that has been proposed by other people, and you can find it in many, except for this thing that is in Shanshan's thesis of choosing T, that's a new ingredient. What I'm going to do is I'm going to show you one application of this mechanism to a data set of biological shapes. And in fact, I will find a refinement of the method. I will show you a refinement of the method that does even better. And the method itself, although I'm going to show you application to only that one data set, I believe is again much more generally valid. And in fact, I am now working with people at the Simons Foundation, at the Flatiron Institute of the Simons Foundation in New York, to apply it to other data sets. But I'm not showing results of that because it's not mature enough. Okay, so this biological, where do the data sets come from? Well, it all started really with a discussion with Jukka Jörnvall, who is a Finnish evolutionary anthropologist, and Doug Boyer, who was at that time his postdoc and who's now a full professor in evolutionary anthropology. Serendipitously, he got a position at Duke, and so that made our ongoing collaboration, of course, much easier since we're at the same place. What they do is they study bones and teeth of mammals, mostly primates, and both animals still living or fossils of animals that are extinct. And they are, like many biologists, interested in evolution. And of course, evolution is something of which we learn a lot through the genome and studying genome evolution. But evolution, the pressure of the environmental factors on the organisms doesn't happen directly on the genes. The genes are the mechanism by means of which traits are transmitted to offspring, but the fitness of the organism is something that happens macroscopically, unless you talk about bacteria, but we're talking about mammals here. So they want to, in parallel to everything they learn from the genome, they also want to learn and see from the organisms. So for that, you need to be able to say, this looks more similar to that than either of them looks to that. So you need to be able to find distances on these data sets. Oh, yes, before I want to make sure that I name everybody, and I'm always running out of time, so if I put that at the end, then I might not get the chance. So these are people who've worked on this over the years. Jaron Lipman was the first person with whom I worked, with Jukka Jörnval and Doug Boyer. He's now a big shot professor at Weizmann Institute, and these actually these are people with whom I'm working on right now at Duke. Okay, so the first thing that I did was they had a project on the complexity of teeth. And in fact, what they did, so we're talking now 15 years ago. So Jukka Jörnval told me when he heard I was a mathematician that he just had, and this is of course a big achievement in biology, he had a paper in Nature where they found, where they published a finding that they could identify the diet of mammals from the complexity of the teeth of their molars. So we're not talking about wear on those teeth, we're just talking about how evolution has shaped the complexity of the molar surface, the chewing surface of the molar. And it was so precise that they could distinguish insectivores from other carnivores, or among the herbivores they could distinguish folivores from frugivores. And not only that, but it was a method that was independent of the size of the animal. It worked for shrews and it worked for elephants. And so that's very strong, because if you have something that works so well kind of universally on a large group of different species, then it's entirely reasonable to assume that the same inference will hold in earlier times, so you can use it on fossils where you don't know what they ate. So that's why was it so important. But he thought I would be interested, and I was interested in how they measured the complexity of teeth. And he said, well, we discovered that there are these engines that computer scientists build to visualize on a screen 3D surfaces. And at that time, if you had used an engine within MATLAB, for instance, it would give you an impression of what it would be if that finely triangulated surface was illuminated by 3 lamps, a blue and a red and a white lamp. And he said, so what we do is we take that and we take shots. We take a shot from the top and a screenshot, and then we look at all these triangles and we look at the change in color from blue to red and so on. I said, so what you're really measuring is the change in the normal to the surface as it moves. He said, yeah, I guess. I said, well, there exist methods to compute that. You don't have to take a screenshot and then photograph it and start counting. And so that's what Jaron developed. And Jaron had just arrived as my postdoc, and he was really excited because in computer graphics, most of the money and the motivation to work on it comes from computer games or video games. And so here he was going to work on a real science project, and he loved it. So he developed, I mean, it was not off the shelf. The notion in mathematical notion, Dirichlet normal energy was an off-the-shelf notion, but then implementing it so that it worked well for these surfaces took some work, and but it worked very well. It worked better than the counting of and so they were ecstatic, and then they came up with a more interesting question. This is something I have found very often in interdisciplinary collaborations, that the first project is not quite off the shelf, so you need to put in some, but if you get a good answer for that, then many more interesting projects open up. So they asked us to find a way to compute cross distances between surfaces without landmarks. So let me tell you a little bit about the data. So the way they work with many small mammals. So they do micro-CT of the teeth, and then they extract surfaces. Now, teeth, of course, are three-dimensional objects, and they have internal structure. Nevertheless, we only work with surfaces because many of the samples that they work with are indeed fossils. And those fossils also have a well-preserved three-dimensional structure and so on, but fossils are often much more precious than teeth of still living animals, if the animal is not endangered. And you can't just say, can I get your tooth, and I'll scan it and I'll send it back by the mail. And so Daubechies has made a live project of it, of whenever he goes for field work, go visit all the local musea and make imprints in resin, very fine dental resin that dentists use, of all the fossil teeth in which you might be interested. But so that means we have surfaces, not the 3D structure. That's why we work with surfaces. So those get then very finely triangulated from the CT data, from the voxel data, and get rendered. And here, one way of rendering them is to actually putting a texture on them and see how it organizes on the tooth. Okay, so typically when they do a study in which they want to study a whole collection and see how different environmental pressures have shaped different different portions of the tooth, is in this whole collection, they have enormous domain knowledge. So they know what points are corresponding to these on the different teeth, and they put landmarks. So they survey their collection and say, this particular feature is present everywhere. Let's put a landmark, and of course they do it digitally, but it used to be done with a machine and so on. And so they put a number of landmarks that are shared by the whole collection. And for each of those, they look at their spatial coordinates. And they then define a distance. So these coordinates, these 3K coordinates of these K points now will serve as a proxy for the service. And here's, for instance, a collection that was used for a study in which all the landmarks are pointed. But so that you have a correspondence. This yellow point is the yellow point, corresponds to that yellow point on all these teeth. And I would not be able to do this. I mean, because I see many more bumps than they find are interesting to label. And to know which bump corresponds to what bump, I mean, I wouldn't know, but they have the domain knowledge, and we respect that. So then you have, but of course landmark placement is time consuming. How do you define the distance then? The distance, so you have K points here, K points there. You find the rigid transformation that puts them in closest correspondence to each other. And that's very simple. I mean, that you can do that via PCA or singular value decomposition. And then you take the sum of the squares of all the distances of corresponding points, and you take the square root of that sum, and that's the Procrustes distance. But placing Procrustes points, these landmark points, is tedious and very time consuming. It's also sometimes a fossil is not a complete tooth. Well, whatever is missing on that tooth, you cannot use in any of the teeth in your collection because you have a missing landmark. You can't put landmarks if they don't occur on all of them. And indeed, you need to have a high degree. So so another thing that Daubechies has done is he started Morphosource, which is a place where many biologists, and not only biologists, also archaeologists, load up data and then make them available to other people to study. So people might be interested in mollusks and have a hypothesis that they want to test, not just on mollusks, but on other biological specimens. Well, it would be hard to do if they don't know how to place landmarks on the teeth of lemurs, for instance. And even there's sometimes cases where even experts don't agree. So this is how the Procrustes distance works. And the question they asked us, look, this distance works well for us, but it's a nuisance. And typically, these are people with PhDs, and they have to spend time putting points on I mean, they do it until their eyes glaze over, or in practice, until they have enough, marked enough that when they do statistical study, they get their p-value to a level that is publishable, and then they stop. I mean, but they have much wider connections in their drawers. I mean, it would be great if those could be used. And if they had automatic methods, then maybe they could be used. So they said, could you? I mean, would it be possible? And we said, well, mathematicians have defined tons of distances between surfaces. He said, yes, and for all the ones for which a software exists, we have tried and it doesn't work. I mean, Gromov distances, it just doesn't work for us. I mean, sorry. So we said, okay. And so we've been working on this since then. So this collaboration started over 10 years ago, and it has gone through many stages, and I'll mention some of those as we go on. So our data, we start with very finely triangulated surfaces. I mean, typically we have between 10 and 30,000 triangles in these surfaces. They already have used computational geometry programs in order to clean them up, and we eventually we want to go back and work with data that come from the micro CT, not have to go through all the cleaning up, because that may introduce artifacts as well, and they would like that, but we're not there yet. Okay, so the first thing we did is we defined two different distances. And one that was inspired by their Procrustes approach, and one that is we had I had much higher mathematical hopes of, because it's pretty. But let me go through them quickly. So what we did for the first distance, so it's called conformal. It's the Wasserstein neighborhood distance. So where we use an optimal mass transport. The first thing, we worked with teeth. I mean, but we can do it also for sphere-like surfaces, but our surfaces were the surface of the tooth above the gum line, and that's a disc-type surface. So for these disc-type surfaces, can all be flattened, can be by conformal transformations, be transformed conformally into the disk. Now, if you work with computational algorithms, then you have to be careful, because if you take all those triangles and you change them conformally into triangles again, you can't do that. I mean, you need to give a little bit, and in fact, what you do is you take midpoints of the sides, and you take those triangles, and you transform them conformally, and then you link things up again. What that means is that some points sometimes get split into 2 points, if you think it through. But so conformal algorithms exist, computational algorithms exist to do that. We conformally flatten, not because we think conformal is so important for biology. Some people have argued it, and biologists object to that in many cases, and certainly not for these teeth, but because it's much easier to search for geometric correspondences from 2D things than 3D things. So we flatten conformally, and we then get a conformal factor on the disk from that flattening, and that you can interpret as landscape. And so you can then do a mass transportation. However, you have to be careful, because conformal flattening is not unique. Each one of these landscapes can be transformed by a Möbius transformation and would be an equally good conformal flattening of the original. And if you do that, then you have to make sure that whatever you define as a mass transportation is invariant for that. So what we did is, so when you do mass transportation, what you have to do is you have to find a distance between points on one. You have to say, how similar is this distance here? Are these 2 points? And to do that, we define little neighborhoods, and they really should be circles, but I drew them by hand. I wasn't paying attention that neighborhoods in hyperbolic metric are still circles. For each of these 2 points, we can use a conformal mapping to bring them to the center, and we can then take a look at the neighborhood of these. And then the only way there still could be a difference, there still could, I mean, because the conformal flattenings are completely different, is that we have to compare each neighborhood here with all the rotated versions of that neighborhood. And that will give us a similarity between points Z and W that is conformally invariant. So that gives us a similarity for these two landscapes. And we can then apply the mass transportation, that to say, find the best way of the infimum among all the things that give these as marginals. So that's the Kantorovich approach to mass distance. And so we call it the conformal Wasserstein neighborhood distance. The other one, it turned out that the Wasserstein neighborhood distance was computationally very heavy. So we said, well, let's try to do a continuous Procrustes distance. But remember, with continuous Procrustes distance, so what we said is, if you have a correspondence, which was given by expertise in the Procrustes case, but we don't have expertise, we just get two data sets. But if you have a correspondence, then what you do is you look at the distance between each point and its corresponding point on the other one, and you integrate over the surface, and you try to find the best rigid transformation from the original to the transformed one to minimize this. So here this is illustrated. Here this is illustrated on this example. I have two curves, and I've built a correspondence. I give you a correspondence which links points. And then with the same correspondence, here I find the rigid transformation that makes the correspondence give the smallest distance. But that's not the best possible correspondence, because in fact, if I took a different correspondence, then the sum of squares would be much smaller. So you have to want to make the infimum over all possible correspondences. But that's an enormous collection. Then in practice, if you implement it, what you do is you put a discrete number of points on one surface and a discrete number of points on another surface. And you try to find now, you want to have these points well distributed. What does it mean? It means that you pick an algorithm so that if around each point you look at its little piece of surface that's closer to it than to any other point, it has about an equal share of the full surface. That's to say, you want their Voronoi cells to have all more or less the same area. But if I do that on this surface, and on that surface, and I now am going to map points here to points there, then what I'm really doing is identifying little Voronoi cells with Voronoi cells, all of the same area. So what I'm really doing is making a discrete approximation of an area preserving map. Okay, so we said let's minimize, let's take our collections, area preserving maps, and let's not take pathological examples, let's take smooth area preserving maps. Biology says, well, with area preserving, we don't believe in that. They said, wait. So that's at least mathematically something, and you can prove theorems about this continuous Procrustes distance and so on. But it's still an impossible collection to search. We were doing computation. So, however, you can prove that if two surfaces have a small continuous Procrustes between them, then the minimizing correspondence is also very close to conformal. And the set of conformal correspondences is something that only has 6 parameters. You can search that. And ultimately, we're not going to trust the large distances, remember? I mean, because we're going to use this diffusion mechanism. So we're happy with something that only gives the right answer when things are small. The other ones we're not going to use anyway. So that's what we did. So that's the second distance. And so among the collection here, we define this continuous. And it gave us, and we visualized for our biologist friends, these mappings between surfaces. Okay, so before, when you work with biologists, they have been so much burned by cases where people were cherry picking in the data and proved whatever and so on. You have to agree before you do an analysis on what the goal is, how you will define success. And so we had defined 3 ways to define success. One way was to compare distances that they computed between samples with our distances. And I visualize that here by showing a matrix, a lower triangle for their distance, upper triangle for our 2 examples. We had numbered the samples in such a way that things that have labels close to each other are also close to each other in the distances of the experts. So what that meant was that, since we believe small distances are the important ones, that close to the diagonal, we wanted to be similar to what they had. And to my chagrin, it turned out that the continuous procrustes distance did better close to the diagonal. You see these 2 blow-ups than the conformal Westerstein neighborhood distances. Other ways we had of comparing was to say, look, let's take landmarks and propagate them, and then compare them with the landmarks that experts would put. And so by all these measures, our methods did, especially the continuous procrustes distances, very well, and so we adopted that one. And they were actually really surprised, and we were gratified that it did so well. Okay, and so this is an illustration of what we do. We take these conformal pictures of 2, and then we conformally change so that they're as close to each other, and then we look at big points, and that gives where they are, and we have the corresponding landmarks on the other one. This is actually one in which we didn't do so well, because this point here should really have ended here, and it ended there. But in any case, it gave us these mappings between them. And the wonderful thing that happened is that this is when we started to convinced the biologists that small distances are the ones that matter. If we took the continuous procrustes distance of a sample, so they prepared for this study a sample of 50 different teeth corresponding to 5 genera of lemurs, and in each genus we had 10 species. And the different colors here show you the different genera. And so they saw this is, of course, the distance map you get is not a 3D, but you do multidimensional scaling, so you find the best 3-dimensional projection of that. And they were gratified to see the amount of clustering here. We weren't happy. I mean, we felt that this was poor clustering. But if we then applied just to those distances, now I'm not using anything else, the distances we got from continuous procrustes, and we applied the diffusion paradigm to it, in which we trusted small distances much more than large distances, and we computed the corresponding diffusion distances. So the paradigm that I explained earlier, this is what we got. And this is when they started believing us when we say small distances matter more. Because that picture clusters so much better than this one did. But nevertheless, at this stadium of the collaboration, so our collaboration works, I mean, whenever I collaborate with people in a completely different field, it starts with a conversation where I identify a question that might be of interest, and I say, if we can do that, would that be of interest? They say yes. I said, well, would you be willing to then work on this? And work on this means not just cheer us on, but have regular meetings where we give them intermediate results and we discuss them with them, and we take their guidance and they learn stuff with us. I mean, unless there's that investment of time from the other party, I don't think it's worth it, because it doesn't make sense for us to take a question away and come back 6 months later with an answer and they say, oh, but that's not really of interest to us, because they forgot to tell you something that's absolutely basic. And they weren't malicious, they just didn't tell you something that everybody learns in kindergarten. But you went to a different kindergarten. And so, so we met all the time, and they kept telling us, but you know, you have this correspondence between this tooth and that tooth, and we don't like. And And I was saying that, yes, but those are teeth that are far distant from each other. It doesn't matter. So then they wanted to propagate through minimum spanning trees, but those, of course, are so that only small distances would define correspondence. But those are very prone to all the problems of minimum spanning trees. If you introduce a new thing, then all of a sudden your tree changes and your correspondence would change, and they didn't like that. And it took me a while, as we were not making progress, to articulate what the problem was. I said, look, you asked us originally a question about distances, but you really care much more about the correspondence maps, don't you? I said, I guess that's true. And so that changed the whole, and then we started making progress again. And in fact, my graduate student, once we had that point of view, Tingwan Gao, who now works for a startup in Chicago, came up with an additional way of defining distances that led to this graph. And this graph they were absolutely excited by, because first of all, the clustering is still a lot better, and it actually, well, I'll come back to peculiarities of, well, maybe I can tell you now. I mean, you'll see the slide later. What happened in this is that 2 of the 5 genera come here, 2 others of the 5 genera, and 2 others here, and there's 1 and so on. It turned out that among the genera they'd given us, 2 of them were frugivores, they eat fruits. 2 of them were foliivores, they eat leaves, and 1 of them was insectivore. And those were really nicely separated this way. And given that we were looking at molars, it's not so strange. So we were really getting strong biological signal. Okay, let me tell you how we got that picture. Well, we used the local distances and made this spectral parametrization that gave us a distance. But in the process of, so we had these teeth and we had distances, but we also had implicitly, not so implicitly, explicitly defined as a tool in between these correspondence maps. And we were throwing them away. After we had them, we computed the distances, and we said, well, you can maybe do much more. In fact, we have really a manifold that we want to learn, and each sample on the manifold is one of these teeth, but each tooth is a 2D surface. So it's a fiber bundle, the manifold of manifolds. And not only that, but we had, for these fibers, we had correspondence maps. And so, which is called a connection in fiber bundles. So, and that's then what Tingwan Gao did. He used this to define, and I'll show what he did. Let's a very, very quick review of fiber bundles, and if this doesn't mean anything to you, don't worry. We'll, so in a fiber bundle, mathematically, what you say is you have an enormous manifold in which points have 2 different labels. One label relates to the base manifold, and one label relates to where they sit on the fiber. So you think of a skin with hair growing of it, and your enormous manifold is all the little pieces of hair everywhere, all those little fragments. And you then, in order to bring down to the manifold, you have a map that goes from the full thing to the base manifold, where on the skin is your hair attached. And they're all hairs, so there is an ideal hair. That's the fiber manifold, that is of which you have copies. And locally, it's just a little product, this times that. Globally, it need not be the same, because locally you can have this Möbius band, and here are examples of fiber bundles that differential geometers like to look at. I mean, here you have segments of tangent lines. Here you have unit, the things at a unit distance on the tangent plane from the tangent point, and so on. So we have a similar situation. And what you can then do is you can, if you have a fiber manifold, you can, and a connection, then you can study diffusion on the base manifold by seeing what the connection brings it to on the fibers. But we don't have a base manifold. But we have fibers, all these teeth. And we now have a connection because we have this map. So what we can do is look at the enormous collection of all these teeth together. And we have a way of diffusing on each of the teeth. And we have a way of diffusing from one to the next. And the mapping takes us around. And in fact, morally, what that means is we have a whole lot of copies of what the diffusion would be on the ground manifold, but it's all very noisy, of course. And you can then look at eigenvectors, just like we did in the diffusion case. We can look at eigenvectors of that. And you look at the eigenvectors, and the eigenvectors have pieces that pertain to the different original fibers that you looked at. So here is a first result that we had. So after Tingran did this, he said, I have this huge dimensional space. Let's take, I have spectrally labeled it. I have done this dimension reduction. It's still huge. Let's take 13 clusters. Let's do the best clustering, k-means clustering, and take 13 regions. First of all, the immediate thing you see is that every tooth has all the regions represented. There was nothing that guaranteed that, but it happens. And the regions don't have the same area. Some places, this little blue region is much smaller than in some others. Look at these two. So even though we started with the continuous procrusted distance, which was really an implementation of area preserving maps, after all these transformations, we're no longer area preserving. And big sigh of relief from biologists because they hadn't trusted it. But this they immediately wanted to use. Oh my God, we can use this for statistical analysis. But hold your horses, we're going to do more. We'll use this already. But it was a first, not only because the areas were not constant, that was not enough, they also really were biologically very meaningful to them. And so that meant they had now a tool, a computational tool, to do things. Another thing is you can try to, from this, try to embed the geometry that's present in this whole collection in a lower dimension. And what Tingran did was he took all the pieces of eigenvectors, of the first k eigenvectors that pertain to the first, and looked at their little gram matrix. So this is a k by k matrix. Now here he's actually showing he doesn't need the same number of points on all so even though he has fewer points here, he can make a k by k matrix out of that. And another one. And now he has a whole lot of k by k matrices. So here I made these k by k matrices with the spectral representation. And then you have matrices, and you can take a distance in matrix space of those. And that's how you define this horizontal this horizontal diffusion distance. And as I said, when these things clustered like that, at the first thing the biologist says, oh my God, maybe you see the evolution tree in there. And no, of course you don't. I mean, we're looking at a molar, for instance. But because you see, for instance, purple and blue are the furthest apart in the phylogenetic tree. But they're both frugivores. And so that's what I was telling you. So they were really excited. Another thing is that if you take the first 3 eigenvectors for all the surfaces, that gives you every point now on each of these teeth is given by 3 coordinates. You can visualize that in 3 space. And this is what you get. You get that you get a surface, well, not so surprising, the fibers are 2-dimensional after all, but you get clearly the same surface sampled different ways. Very nice, we've identified the fiber. This allows us to do a better registration. Because of course we chose 500 points on one tooth and 500 here. None of those points were going to truly correspond. We tried to find the best correspondence, but they're not truly corresponding. What we now can do is we can say, oh, look at this red point here. There are 3 blue points next to it. On this representation, we can find a weighted, the barycentric coordinates in that triangle of that blue point, of that red point in the blue triangle. So that is what this red point really corresponds to. So we can now define a consistent mapping that's coming out of the analysis of the whole of the fiber bundle. So what this does is it gives us a way of denoising. I mean, of course, we have made this assumption that we have this correspondence between all the fibers, that this is the true mathematical structure. But by imposing and recognizing, we can then denoise lots of the other stuff and get a nice correspondence map, a registration everywhere. And then you can redo the whole thing and gradually learn your fibers best. And once your correspondence is something you're really happy with, I didn't talk about you. When we did our diffusion, we built our diffusion locally, like a little Trotter product, because locally it's a product of spaces. So you diffuse in one and diffuse in the other. Now the problem is we don't really know the right time to diffuse, with the ratio between the 2 times. You have some leeway there. So what we can do is we can first learn the fiber and the connection, and then we can play with that in order to start feeling more of the manifold, the base manifold in the diffusion. At first we diffuse out over the base manifold, so we take really an average. And then we diffuse less on the base manifold in order to get a better local feeling. So we're working with that, and that is not finished. But so, oops, I'm sorry, I had, I took slides from the wrong talk. I mean, I had some other slides where this was explained. So other things we are doing, for instance, is doing using Gaussian processes to do landmarking. I mean, we infuse, I mean, so at where we are, what I just described is where we were a year ago, we are now exploiting different methods to learn more about this correspondence map and bring it more to concepts and parameters that our biological collaborators can exploit. But the interesting thing is that we started with these continuous procrustes maps. We were so proud of them at first, 10 years ago. Now we call them the crappy maps. CP for crappy, because they're the maps that help us to get a much better registration later. And in every one of those intermediate steps, we have found strong biological signal, and which is what was important to make sure that we were not being led astray. But we also have found an approach that really works well for data sets, high-dimensional data sets, where each data point is a structured thing. Now, this happens a lot. When you want to apply diffusion methods, it often is because you judge that this is more similar to that than either is to that. How do you find that out? Because you look at the structure. You say, this looks more like that because compare these two. Well, if you have that, then you can apply this method. And you've seen what a difference it made to keep the correspondence map going for longer. And I'm going to stop there. Thank you.
Thank you very much for a wonderful talk. Are there any questions?
Well, first of all, thank you very much for your talk. Regarding the, my question is the following. Are there any circumstances, any perhaps biological data sets in which it would be more convenient to consider a hyperbolic diffusion operator instead of parabolic diffusion operator, maybe associated to the relaxation time lag or something of that sort?
I'm happy to consider one, and it might be of interest. We really are exploiting peculiarities of diffusion in Euclidean spaces in building our kernel. I don't know whether I could do that if I do work with hyperbolic diffusion. So I don't know. My answer is I don't know, either biologically or I don't know how to do it computationally, actually.
Thank you.
Thank you for your talk. Regarding the connection, is there any relevant information arising from the curvature of this connection?
Okay, so we hope so. Right now, we're looking at species that are closely related and for which in the not too distant past, they believe to have a common ancestor. And the changes in the tooth surface are such that we believe the whole situation is the curve. It's flat. The maps are commutative because you can go back to a common ancestor and come down again. And so going from A to C via D is the same as going from A to B and B to C. But it's clear that that's not going to continue to be true. If you look at teeth among mammals, I mean, seals have teeth that are so different from what primates look like. So what we believe is, and we don't have a good way of doing that, is that we will need to coarse grain our fibers and coarse grain the connection, so that, and that the data set itself should tell us how to do that coarse graining, so that we would still have commutativity if we go back for that coarse grain structure to further ancestors that are further apart. So we want a data-informed way of doing that coarse graining, and so the curvature would come in there. So we would get non-commutative structures that we would find the curvature, and we don't expect there to be, on the things we look at, necessarily global curvature. We believe small curvatures that will need to be flattened out by the coarse graining in order to keep the commutativity going. And absolutely, there needs to be a way of doing that, because there will be curvature if we have a very large, very diverse data set in these teeth, and we need to deal with it. But it will be governed by the fact that we assume there to be a tree structure behind it.
It's also kind of a biological landmark then. It tells you how far apart specimens
exactly.
are.
Yeah.
Thank you.
Thank you. That was really nice. So these were all species of different genera, but all lemurs, right? So these are not other types of insectivores like
no,
bats
for the
or
moment
whatever.
we're looking. When I showed you the 50, those were all lemurs. When I showed you the comparison of the matrices and so on, there was much more variability there. We had primates and non-primates there. But,
Okay.
but for the fine-grained stuff, it's all lemurs.
Right. So you haven't done this level of analysis between different genera or
well, between
widely different.
Yeah, between different families. No,
different families.
not yet.
Okay. Okay.
But and I believe that at that point the curvature thing. Well, I mean, I believe in order to keep doing what we're doing, we need to then first coarse grain at the level of the fiber and of the connection in order to make things work.
Okay. Thanks.
There was a question at the back too.
Hi, Professor. Really nice talk. I'm a huge fan of your work, especially in wavelets. But anyway, In a few slides, you have different pictures of many teeth. So that makes me think if there is any chance that you can work on shape changing surfaces. I'm thinking about plasma physics, for example, that change over time.
Yes. So there might be, but I think so. First of all, what we're really doing is study on shape space. I agree, and so it could be used for other things in shape space, and certainly a shape that is evolving could be analyzed this way, and one could see in its parametrization how these parameters change. I think that definitely would be an interesting application. For some surfaces that occur in physics, I think more in statistical mechanics, but maybe also in plasma, certainly in astrophysics, you have roughness to those surfaces. And I think to analyze those, you need different parameters from what we're doing, but I'm not sure about that.
Thank you.
Any other question?
I have a question.
Yes.
Have you analyzed human molars?
So we could put, and actually in the data set of where we compared this, there were some human teeth as well. I think they really try to I mean, so there's two things when you say how do you analyze human molars. There's one, the medical thing or dentistry, where they really want to have things that are very close. So they have ways of trying to find whether a molding is identical to the tooth and so on. And so these are tiny distances by us, and they use procrustes. I mean, the other thing is at human evolution. If you want to look at hominids and Homo, present Homo, I don't know whether, I mean, I know Doug Boyer is very interested in analyzing those, but as well. But there, the one work we have in progress is not about teeth, but others. We're looking at wrist and heel bones. Over in different precursors of Homo to see whether we can pinpoint changes between the more quadruped way of motion and biped. But we haven't found anything that's really, we haven't hit a home run there, let's say. So, but yeah, I mean, but that's really what ultimately these once we have the mathematical method really under control and can guarantee, look, this is what it does, and so on, then of course the biologists can run with it. And but we've been developing this tool to.
Be possible to know if we are carnivores or herbivores?
Well, I think biologists are all agreed that we are omnivores, but so we eat both. But and that's what our teeth show. The complexity of our teeth shows that. But of course, that's what we evolved to. I mean, we can then decide to do other things.
Thank you. Any other questions?
Yes, thank you very much for this great talk. I wonder if this work of yours could be used to uniquely describe 3D surfaces in an efficient and, let's say, building a compact code for describing surfaces that is unique to, that is unique, you know what I mean? Making codes for describing a surface.
Well, I think you could build, there are lots of surfaces for which different approaches exist and are better. than what we do. In computer graphics, as I said, computer graphics is governed by video games. So they have surfaces and ways of comparing surfaces and mappings and so on. And typically their surfaces have much more features, many more features. I mean, when we first told them about these teeth, I said, those don't have any features at all, because they're used to a monster aardvark attacking a dinosaur or whatever. So when you have big features like that, you have very different ways of approaching it. You try to find local, almost isometries between pieces of the surface, and so there is a lot of very beautiful mathematical work that exists there that doesn't apply here, and I don't think that what we did would apply there. I mean, it's really adapted. So I think there are two strands to what I showed. I think there is a way of extending diffusion maps by using the fact that you have structure in your samples, and we don't typically exploit that. And this shows that it's useful. So that's a general thing. But the particular thing that we did, and the particular way we started from these continuous progressive distances, so that's really adapted to these particular surfaces. And but you could apply the same thing to start from a different surface to begin with, and then do this analysis. And it might be useful, I don't know.
Well, first of all, a beautiful lecture, but would it be a topic of interest in understanding these mappings or these analysis methods for not characterizing previous or extinct or extant species, but how these species would evolve and how in this case, the teeth would change?
So you're thinking of doing it, go to the future?
Yes.
Yeah, I don't know. I mean, that again takes things back to the biologists. I mean, the idea is we wanted to build a mathematical tool that really, at every stage, was tracking things that were biologically meaningful, because I don't believe in having a tool where the biological content drops out from the sky if it's not there at the intermediate stages. But then what you do with it. I mean, we didn't do any analysis about this is what happened in the environment and that's why it changed this way. We just were we were trying to build, based on mathematical understanding of what is believed to live in the data set, tools to extract that mathematical understanding from the data, so that they could then do things with it. But then once they have those parameters, they can do statistical analysis in order to relate it to change in the environment, for instance, and so on. And once they understand that, they could try to take that to evolution to the future. But those are not things, those are, first of all, not yet done. And it would need a couple of other stages before they could be done. Does that make sense?
Thank you.
En el salón C13, pero no sé cómo.
Hay una pregunta en el salón anexo de donde estamos transmitiendo la conferencia. No sé, Mauricio, si nos pueden conectar el sonido, por favor.
And I've said several times we're building a tool, but I don't want to say, oh, we're only building a tool. I mean, and they have to do the science. Building the tool incorporates a lot of science. I mean, we have to understand how they look at the science in order to build that tool. I mean, to make it mesh nicely. So that's one of my experiences. You have to really understand the application, or enough of the application. I mean, I can still not name landmarks on teeth. I mean, I can fake it. I can say, oh, that's an antoconate. But I mean, everybody will laugh at me who knows what this really means. But build, it's not just ancillary work. it has interesting mathematical questions like this coarse graining, which I don't know how to do yet. I have to think mathematically about how will I do this, and curvature will play a role in that. So I don't want to belittle building the tool. I mean, there's lots of really interesting mathematical questions, and the interesting thing is that some of the mathematics then can lead to other stuff. So I think this double diffusion approach on this fiber bundle is something that I think some pure mathematician will make much nicer than what we have done. But
Professor, there are,
yes. Oh, sorry.
there are another question.
Oh, yes.
Adelante, por favor.
Excellent presentation. Thank you so much. I was wondering if, have you ever applied this kind of work of like the reduction of landmarks in other projects? Because I recently see like another tag of yours, another talk of yours, and I find it quite interesting because in there you were like reconstructing some kind of art, and
Yeah.
and
So-
in that talk you said also that it was a whole process because you needed to apply machine learning algorithms to reconstruct art. So it seemed quite interesting.
Yeah,
And
so we
that's why I
haven't.
wonder.
Yeah, we
Yeah.
have not used this approach in this other work that I do with doing image analysis to help art conservators. But it's intriguing thought. We are looking at other projects, not in the art thing, because I've recently learned that people in neuroscience use diffusion methods and in order to define distances between the firing sequences of different neurons. And there again, those are time series, they're not surfaces, but they again are looking at things that are more or less similar. And so I think I hope to apply these methods to those. So as I said, I don't think it's linked at all to this particular application, but we haven't used it for art. For images, it's kind of hard sometimes because but we could do it on feature vectors that we define in the art. Yeah, thank you. This is an interesting suggestion. I hadn't thought of it. Yeah.
Thank you.
Any more questions? ¿Más preguntas? Okay, entonces, agradecemos de nuevo a la doctora Ingrid Daubechies por esta inspiradora presentación y a todos ustedes por su asistencia. También agradecemos a quienes nos acompañaron de manera virtual a través de Zoom. Les recordamos que la doctora Daubechies dará otra plática mañana a las 12 en el Instituto de Matemáticas. Gracias, que tengan una excelente tarde y agradecemos nuevamente a la doctora Daubechies.